Welcome to the Organic Chemistry Podcast, Dr. Brian Lloyd's scribblecast of organic chemistry lectures and solutions to homework problems. In this scribblecast lecture, we're pressing on with our investigation into carbohydrates. Last scribblecast lecture, we looked at the oxidation of monosaccharides utilizing Tollen's reagent, Fehling's reagent, and Benedict's reagent. Because these reagents were in base, we learned that the oxidation of the aldehyde group in the sugar produced a CO2-, a deprotonated carboxylic acid. Well, there are other reactions that can convert the aldehyde group and other fragments of the molecule into carboxylic acids. What we want to do is begin looking at these utilizing a D-glucose molecule. So let's draw a D-glucose. Here is D-glucose. And what we want to do is examine what happens if we oxidize different portions of this molecule to produce a CO2H. Now, if we react this with molecule with Br2, H2O, at a pH of 5 to 6, notice that's a slightly acidic pH, we will oxidize the aldehyde to a CO2H. We will produce the carboxylic acid on top. Now we're going to look more closely at this molecule in a little later on in more detail, but the question is, what kind of functional group have we made? We have taken our aldose and converted it to something else. If you oxidize the aldehyde end, carbon 1, and produce a carboxylic acid, you have changed your aldose into an aldonic acid. In this case, D-glucose becomes D-gluconic acid. Okay. Now we can use different reagents. For example, we can hit this with HNO3, nitric acid, and heat. If you use nitric acid and heat, you not only take the aldehyde to a CO2H, but you convert the terminal CH2OH into a carboxylic acid. This molecule is referred to as an aldaric acid, and D-glucose becomes D-glucaric acid. So, if carbon 1 becomes a CO2H, you produce an aldonic acid. If it's D-glucose, that's D-gluconic. If you treat with nitric acid and oxidize both ends of the molecule, you produce the molecule D-glucaric acid. It should be interesting to note what would form if we had used L-gulose instead. I'll draw it here. What if we had used this molecule? Well, this is L-gulose. L-gulose in nitric acid would have been converted to L-Gularic acid. An aldaric acid, and this is L-Gularic acid. Now, interesting, let's rub out L-gulose here. I want you to compare structurally L-Gularic acid to D-glucaric acid. Look very carefully at them. Notice that if I rotated this that way, swung this up, this OH would be on that side, this OH would be on that side, that OH. Just rotating the molecule turns D-glucaric acid into L-Gularic acid. In fact, these two molecules are identical. They're the same thing, just spun around. Why is that the case? Well, notice when both of these groups, here and here, become identical, which one at the top is at the top doesn't matter. You can rotate it. And so the molecule D-glucaric acid and L-Gularic acid are the same thing. Now, this can happen sometimes in reactions when you put identical groups at the top and the bottom. There are actually two possible names. All right, and it's perfectly fine to call it D-glucaric acid, and it's perfectly fine to call it L-Gularic acid. Fascinating. Okay, let's take these two molecules now and remove them. There is one other type of sugar acid that's very difficult to make. And what is that? Well, it turns out, what if you want to hydrolyze this, oxidize the terminal CH2OH, but not the aldehyde group? How could you do it? Well, it turns out to not be a very easy process in the laboratory. It actually requires you to put on something called a protecting group, and we'll be looking at protecting groups later. But it does turn out there are biological enzymes that can do this. The reason it's so difficult in the lab is the aldehyde is more easily oxidized than the alcohol. But there are enzymes that will only bind to the terminal CH2OH, and therefore they will only act on that, and the aldehyde stays intact. If the aldehyde stays intact, and the terminal CH2OH becomes a carboxylic acid, you have generated a uronic acid. A uronic acid. And D-glucose has become D-glucuronic acid. Now, what's really neat about this is it's fairly easy to do this reaction, whether you have the open chain form of D-glucose or you have D-Ribopyranose. If, for example, and I'll just rub out the name D-glucose, give myself a little more room here. If I draw the alpha, Haworth of alpha D-Ribopyranose, and I do this reaction on it, so I carry out the reaction with enzymes, and I oxidize the CH2OH, Well, the product's very easily to draw. The terminal CH2OH, which is pointed up, has simply become a carboxylic acid. Now, this molecule right here is called alpha D-Ribopyranose. What's interesting in nomenclature is that once you form the uronic acid, this molecule here just becomes alpha D-glucuronic acid. There's no mention of the pyranose, even though it is in a pyranose ring. You know you must draw the pyranose ring because of the presence of the term alpha. So if you see alpha and you're asked to draw this molecule, you know you have to draw this because of the presence of the alpha. However, if, and I emphasize this, if you look at the name D-glucuronic acid, you know it's the open chain form because there's no alpha or beta in front of it. It's interesting that the term pyranose is dropped. Lastly, notice the similarity between alpha D-glucopyranose structurally and alpha D-glucuronic acid. Students often make a mistake because I'll give them the uronic acid, but they'll glance at it and they won't notice the difference between the CH2OH and the CO2OH. Those two structures are very similar, and if it's drawn and you're not paying attention, you might just see the CO2H but think it's a CH2OH and call it alpha D-glucopyranose, and that's wrong. It would have to be named alpha D-glucuronic acid. So watch for that. Okay. Now let's go back and look a little more closely at the aldonic acid. Because the aldonic acid can do something a little more complicated once you've formed your product. So remove this. I need a faster eraser. That's what I need. We're getting there. There we go. Okay. So let's take this back to a, oops, change that back to blue. CH2OH. So this is our aldonic acid. So we have our aldonic acid. What's interesting is that the aldonic acid can cyclize. So the first question that I raise is, does it cyclize into a six-membered ring? Oops, that's not an aldonic acid. Well, to understand what's happening here, we have to remember some basic chemistry that we learned last semester in alcohols. That is, a carboxylic acid can react with an alcohol in acid to be in equilibrium with a functional group that we call ester. A carboxylic acid can react with an alcohol to make an ester. Now, this long molecule has alcohol groups that can wrap around and attack the carboxylic acid end of the molecule and make an ester in a ring. The question is, what size of ring is optimal for a ring containing an ester? We would first think that the six-membered ring, like in the Pyranose case, is the ideal ring. The difference is, if you make a six-membered ring where you have an oxygen and you put a double bond O in, you have one carbon that's sp squared here, right here. And remember, six-membered rings are ideal if you have sp cubed carbons. It turns out that yes, you could form this, but it's not quite as stable as if you make a five-membered ring. And so what forms is a five-membered ring system that has the ester. Now, esters in rings are called lactones. If your ester is in a ring, it's called a lactone. And to determine what hydroxyl is used in cyclization, what kind of lactone I've made, I identify the OHs. Here is my functional group, the carboxylic acid. And so the carbon that has the first OH is called the alpha carbon. Carbon with the next OH is called the beta. Carbon with the next OH is called gamma. And then we have a delta carbon. Now, in order to make a 5-atom ring, the carbon with the C double bond O is used. The alpha carbon, that's 2 carbons. The beta carbon, that's 3. The gamma carbon, that's 4 carbons. And then the oxygen of the OH is in the ring. That's 5 atoms in a ring. So the gamma carbon's OH is used. Hence, since we use the gamma OH to make this ring, this is referred to as a gamma lactone. Okay? A gamma lactone. All right. Now, if I cyclize this, I can draw the fissure of it. And I'm going to do that right below the 5-membered ring. So we can draw a fissure of this gamma lactone. So I'm going to do that. And then we can use that fissure of the ring structure. So I'm going to have a C double bond O. I did not use the alpha carbon to cyclize. I did not use the beta carbon's hydroxyl group to cyclize, but I used the gamma. There we go. And then below that I had my CH2OH down here, and I had my reference hydroxyl. So the reference hydroxyl was not used in forming the cyclization. Now, when I draw my 5-membered ring, and this is just a Haworth with 5-membered ring, unlike the Haworth in the 6-membered ring, Haworths with the 6-membered ring had the oxygen in the upper right vertex and the anomeric carbon here. Haworths with 5-membered rings have the oxygen in the upper vertex and the anomeric carbon right there. Now, our anomeric carbon's not chiral because of C double bond O. Okay, so it's not really an anomeric carbon in this case. Now, I can take and put in my groups, and I can put in these two OHs pretty easily because I can follow my rules. In a D sugar, groups on the right in the fissure are below the plane of the ring, and groups on the left of the fissure are above the plane of the ring. Now remember, in a D sugar, the terminal CH2OH is up. That rule applies if the reference OH is in the ring. In this case, the reference OH is not. So I've got a problem. The structures that I put in this position those two chiral carbons, I don't know whether to draw them up or where to put the OHs. So I actually have to figure out the chirality. Now, if I do the chirality for this system, that's one of the reasons I had you do all that nomenclature right at the beginning in the practice problems, where instead of calling D-glucose by its common name, have you come up with the IUPAC name. And you learned that for molecules like D-glucose and D-fructose and that, when the OH is on the right, it's R. So these two chiral centers are R, and I can immediately know that, and you can check that. Now, if I prioritize the oxygen off this chiral center right here, is going to be priority 1. And if I proceed out, carbon, oxygen, I'm going to find this one that's coming out towards me is 2. Now the question is, where do I put the bond, up or down, for the group that's priority 3? And if I want R, 1 to 2 to 3, it actually has to be drawn up. Now this is difficult to show on the Scribblecast, and so I would encourage you to build a model. But you will find when the group 3, the third priority group, is placed up, this chiral center is R. Now, that means the CH2OH is up here. Fine. Now the last chiral center, which has to be R, is this. And we're going to have an H, of course, which is priority 4. I can put the OH on this side or the other side. Well, this is going to be my high priority group. My second group will be this one, 2, 3. And when the H will be number 4. I have to interchange and move number 4 to the bottom. If I do this, that'll put number 2 where number 4 is. 1, 2, 3. That'll make it S. But I did one switch, so that means this structure is R, and we want R. So indeed, having the OH on the left also produces R. And so it was a little tricky to generate the Haworth diagram of the gamma-lactone, but here it is. Much easier to have the Fischer. Now, let's name these molecules. Well, if I name this molecule, remember it comes from the aldose D-glucose. In the open chain form, D-glucose, when it's oxidized, becomes D-gluconic acid. What is it when it's in the Haworth? Well, in the Haworth, it's called D-gluconic acid. When it cyclizes, in the Haworth, or in the cyclic Fischer, it becomes D-gluconic acid Gamma-Lactone. So if you see the Gamma-Lactone, it cyclizes. Now, does this occur for every carboxylic acid? And by the way, you should note the reagent here, of course. What was the reagent? It was Br2, H2O, pH 5 to 6. Now, when we oxidize and get carboxylic acids, can we form these gamma-lactones all the time? In actual fact, yes. With Br2, H2O, pH 5 to 6, the open chain form will immediately convert to the gamma-lactone. And the correct product is actually the gamma-lactone. And you may be asked to draw it in the Haworth or the Fischer. And you have to do that. Likewise, note that aldaric acids could form these cyclic gamma-lactones. And because aldaric acids have 2 carboxylic acids, one above and one below, you'd get two possible gamma-lactones. So because of that, I'm going to keep it simple. You only have to draw the lactones, either the Fisher or the Howarth, the gamma-lactones for aldonic acids. Even though aldaric acids can do it, we're not going to draw it. It's just getting a little too complicated. So I'll let you keep aldaric acids in the open chain form. You only have to draw the final product as a gamma-lactone for aldonic acids. And you need, of course, to know the nomenclature. What about all the reactions that we learned last Scribblecast lecture that included Tollen's reagent, Benedict's reagent, and Fehling's reagent? Well, remember Tollen's reagent didn't leave the proton on. The proton came off, and you form a CO2 minus. When that happens, you cannot form the lactones. You need the proton on the carboxylic acid for the cyclic ester to form. So that's the good news. If I give you Tollen's reagent, or Benedict's reagent, or Fehling's reagent, you can stop at the open chain form. You need to only draw the straight chain aldonic acid with the deprotonated carboxylic acid. However, the bad news is, if I give you Tollen's reagent to step 1, and then I put 2 H3O+, the H3O+ protonates the carboxylic acid. And then it's just like Br2, pH 5 to 6. This carboxylic acid will go to the lactone. There you go. Gamma-lactone. So if you see Tollen's reagent plus H3O+, the correct product is drawing the gamma-lactone. But if you just see Tollen's reagent, then Tollen's reagent will form the CO2 minus. Okay. Now the last thing to note, let's once again visit the uronic acid. Biological systems. So here's alpha-D-Ribopyranose. Will react enzymatically in biological systems. So my alpha-D-Ribopyranose will react and produce alpha-D-glucuronic acid, The glucuronic acid derivative is very important in animal systems because many toxic substances, are excreted as glucuronides. In fact, in plant and animal systems, D-glucuronic acid can be converted to L-gulonic acid. How? Well, once you've got this alpha-D-glucuronic acid, the carboxylic acid, its open chain form, of course, remember, looks like this. The aldehyde end has been untouched. There are enzymes that will take alpha-D-glucuronic acid and reduce the aldehyde to a CH2OH. This hemiacetal, remember, is in equilibrium with a tiny amount of the open chain form. So the enzyme can reduce this aldehyde. And if that gets reduced, I'll just use H bracket for the enzyme that comes in and reduces it, you get a CH2OH up there. And when you do that reduction, your uronic acid has changed. And you've drawn an upside down fissure of an aldonic acid now. And so if I just rotate this, if I spin this around like that, this is the same molecule. So when you reduce the aldehyde, turn it to CH2OH, CH2OH groups are usually drawn to the bottom. So if you spin the molecule around, what is this guy? Well, that's just L-gulose with a CO2H on top, so it's an aldonic acid. So that's L-gulonic acid. Now remember, L-gulonic acids, like L-aldonic acids, can form a gamma-lactone. That is, here's the alpha carbon, the beta carbon, the gamma carbon. The gamma hydroxyl can cyclize. If I draw a fissure of this, I'll get C double bond O, OH, 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 so here's the fissure of L-gulonic acid gamma-lactone. If I was to draw a Haworth of that, I would have, remember left is above, I would have this. Now, this is L-gulonic acid gamma-lactone. It turns out there's an enzyme that will oxidize these positions and turn this single bond here into a double bond. And so what happens is form a double bond here, and your OH are now trigonal planar, and you get something like this. That last, and we'll just put oxidizing here for enzyme, converts my L-gulonic acid gamma-lactone into what is called L-ascorbic acid, gamma-lactone. The L-ascorbic acid gamma-lactone is known as vitamin C. So certain plant and animal systems can produce vitamin C. We have to consume vitamin C. It's necessary. It's a very important sugar acid. Vitamin C is one of these sugar acids derived from glucuronic that is converted into an aldonic acid. And this gamma-lactone form is very important. That's why you have to learn to draw it and name it. Prolonged lack of vitamin C in the diet results in scurvy. Less severe deficiency will produce alterations in connective tissue and may also cause decreased resistance to some infections. It's present in large amounts in citrus fruits and tomatoes. Okay. So there you have it. Sugar acids, Aldonic acids, Uronic acids, and Aldaric acids. Very important chemistry, both biologically and in the laboratory. What we want to do now is look at a reaction that was very important to qualitative analysis of unknown carbohydrates in the laboratory. Suppose you were given an unknown sugar and you wanted to identify that sugar. Now the test I'm going to give you is not able to identify the sugar, but it certainly would narrow it down to what kind of aldose you have. Is it a tetrose, a triose, a pentose, a hexose? That would be extremely useful. Okay. This reaction that we're going to look at is referred to as periodic oxidation. So periodic acid oxidation. Now, periodic acid oxidation, is the oxidation of either 1,2-diols or keto alcohols, or carbonyl with a neighboring alcohol. If you have the diol and you react it with periodic acid, which is HiO4, you will cleave the carbon-carbon bond, producing 2 aldehydes. If you have a keto alcohol, the keto end produces a carboxylic acid, whereas, of course, the aldehyde end produces, or the alcohol end produces, an aldehyde. I should have really drawn this the other way. Okay, now, you should note that if there's anything intervening in between the carbonyl and the carbon with the alcohol, for example, if you have something like this with a CH2 there, you get no reaction with HiO4. So with HiO4 you get no reaction. And the reason for this is the intermediate. When you have a diol or a keto alcohol, which have the functional groups next door to each other, if you react it with HiO4, you lose H2O, so minus H2O, and you form a cyclic intermediate, very reminiscent of what we saw in the permanganate low temperature oxidation of alkenes to make 1,2-diols. And so here we go. And what happens is you end up getting a paracyclic movement of electrons, carbon-carbon bond ruptures, and you end up making 2 C double bond O's. Now, because the HiO4 fragment has to bridge the 2 carbons, if there's anything in between, it's not capable of spanning the distance. So when there's a CH2 here, you get no reaction. Now, I'm not going to expect you to know this intermediate. It just helps you to understand what's going on and why this occurs. However, what I do expect you to know is how this applies to a carbohydrate. Why was this reaction useful? Suppose you were given an unknown sugar, and you took this unknown sugar to the laboratory and you were going to apply periodic acid oxidation to that sugar. Suppose that the sugar you were given was D-erythrose. What would have occurred? Well, let's draw D-erythrose. D-erythrose is an aldose, and has 2 chiral centers, such as this. Now you hit this guy with HiO4. Now the attack will occur at all different sites, but let's proceed systematically and consider what would happen if the HiO4 bridged between the C double bond O and the OH. That would follow this reaction. The C double bond O end would become a carboxylic acid, so you would produce methanoic acid. It would cleave that. The alcohol end here would become a new aldehyde, and you'd get C double bond O, H, OH, CH2OH. You'd get D-glyceraldehyde. Now HiO4 could attack again. If it attacked again, and hit the carbonyl and OH, the carbonyl end would become a carboxylic acid, so I'd get methanoic acid again. And then where the OH is, that would become an aldehyde attached to a CH2OH. Now, if HiO4 attacked one more time, going after the C double bond O and the CH2OH, it would split it, and the C double bond O end would become a carboxylic acid, and the CH2OH would become an aldehyde. When the reaction was completely done, and I had oxidized the molecule completely with HiO4, I would have produced 1, 2, 3 methanoic acids, produced 3 methanoic acids, and 1 methanal. And in the lab, they could have analyzed for that, and they would have found a 3-to-1 ratio between the methanol 3-carbox and the carboxylic acids, the methanoic acid. And the ratio 3-to-1 would have told them that the sugar they're working with was a tetrose. You see, a tetrose would generate, by this method, 3 carboxylic acids and 1 aldehyde. And so if they were given an unknown carbohydrate, periodic acid oxidation, could I help classify, excuse me, help classify the carbohydrate? Okay. Now, having looked at sugar acids and periodic acid oxidation, we need now to examine some other sugars and their cyclization processes as well as reactions that they can undergo. And one sugar that you've already seen is important from the de Bruyn-van Eckenstein transformation is D-fructose. Now, interestingly, D-fructose, when it cyclizes, forms a mixture of not just Pyranose rings, but also Furanose rings. Let's see what I have with. Let's draw the open chain form of D-fructose. Actually, yeah, let's do that in the middle. Might be a good idea. So here is D-fructose. Just like glucose in aqueous solution, D-fructose can cyclize. What's interesting is it does form a significant percentage of 5-membered rings. Now you remember the word Pyranose for the 6-membered ring. Here's our Pyranose ring. It came from the molecule Pyran. Because Pyran was a 6-membered ring with an oxygen in it, the term Pyranose was used for the 6-membered ring that has sugar. Well, what are we going to call 5-membered rings that have an oxygen in them? And they're named after the 5-membered ring with 2 double bonds. And this molecule is called furan. Hence, when you draw a sugar with a 5-membered ring and an oxygen in it, it is called a furanose ring. Now, fructose does produce a certain percentage of furanose rings, even though the ring structure is probably not as stable as the 6-membered ring. We do get some furanose ring. And we can draw this. Notice, to get a 5-membered ring, I've got to cyclize and react with the C double bond O. The C double bond O is my anomeric carbon. So the anomeric carbon isn't always numbered 1. In this case, 1, 2. Carbon 2 is my anomeric carbon. Now, if I want a 5-membered ring attached to carbon 2, if I go down I'm going to get carbon 2 becomes counting 1, 2, 3, 4, and the oxygen will be 5. The reference oxygen is going to cyclize. So I can do that. I can draw, okay, here is this OH. Here is that OH. And now the reference oxygen is going to cyclize, and I'll do that this way. And let's draw the alpha. Remember, alpha means the OH will be on the same side as the reference. So the reference O is to the right, so I'm going to put the OH here. That leaves the original CH2OH, and I'll make that go to the left. And then my terminal CH2OH. Now this is D-fructose. And what I have over here, or I've drawn over here, would be called, because the OH is to the right on the same side as the reference, alpha D-Fructofuranose. Can I squeeze that in? Probably not. There we go. Now, alpha D-Fructofuranose is present in solution to 9%. Okay. I could draw a Haworth projection for that. In a Haworth projection with a 5-membered ring, the oxygen is drawn at the top vertex and the anomeric carbon is to the right. In a D sugar, anything to the right in the fissure is below, so the OH is down. The CH2OH is up. Left is above. Right is below. And since the reference oxygen is used in the cyclization, the D sugars put the CH2OH on the terminal group up. And so there's the Haworth for alpha D-Fructofuranose. Now we can draw the same thing, same type of thing, for the beta. So I have my OH, my reference oxygen cyclizes. I better give a little more room for that CH2OH. Beta, the OH is opposite the reference. That puts the CH2OH of the top of the fructose to the right, and the bottom CH2OH. And so this is beta D fructofuranose. And I'm out of space, so I'll put a hyphen OSE. Beta-D fructofuranose, and its Haworth would be identical to the alpha except at the anomeric carbon the OH is up and the CH2OH is down. And you can get that from the fissure. Right is below, left is above. Okay. And so there's beta-D-Fructofuranose. Beta-D-Fructofuranose in aqueous solution for fructose is present at about 31%. So even though these Furanose rings form, they certainly aren't the majority of what's in aqueous solution. I wouldn't want you to think the Pyranose rings aren't important. It's just we haven't drawn Furanose rings before. And so we could take a look at this for the Pyranose, and we will do that shortly. Note, you could convert these molecules to glycosides. Now, once we've cyclized, what kind of functional group have we formed? Very important to check. Let's grab one of these. Let's look at this carbon right here. That carbon has two singly bonded oxygens to it. So it's in the hemiacetal, hemiketal, acetal, ketal family. Because there is an OH, it's hemi. Okay. Is there a hydrogen attached to that chiral carbon? There is no hydrogen attached to it, so it's in the ketal. So these are hemiketals. Hemiketals and hemiacetals are in equilibrium with the open chain form. They do mutarotation. Everything is the same. But if we wanted it not to mutarotate, I could react this molecule. Okay. I could react this molecule. Well, let's do the beta. Let's react this one with methanol, okay, or ethanol. Let's use ethanol. If I hit it with ethanol and anhydrous HCl, I could react this molecule and produce a glycoside. And the molecule I would get, I'll just convert this one. This is CH2OH. And up above, I'd get the OH here becoming an OCH2, CH3. Now remember, this is a glycoside, so the ose ending is replaced. The ose ending is replaced with oside. And since this was from beta-D-Fructofuranose, this becomes a beta-D-Ribopyranose. So I have beta-D-Fructofuranoside, and the only thing I have to indicate is what's attached to that oxygen. Well, we have an ethyl, and that comes out front as a separate word. Ethyl beta-D-Fructofuranoside. Okay. So if we want to prevent mutarotation, we can produce glycosides of both Furanose rings, as we did in the case for glucose with its Pyranose rings. I would like to point out also that, and you should note, that the Furanose rings only make up about 40% of what's in aqueous solution. There are also the Pyranose rings. So let's rub this out and let's draw some Pyranose rings for this. And take a look at what that looks like. So if we rub this out, we can leave the Furanose rings up there. The open chain form, if it's cyclized to make a Pyranose ring, would need to produce a 6-atom ring system. Now if we start counting at carbon 2, 1, 2, 3, 4, this carbon's number 5. We have to use the oxygen of the CH2OH. Okay, so if I do that, I'd have to generate something like this. where the oxygen from the CH2OH cyclized. Now, if this one was beta, the OH that forms would have to be opposite the reference. The reference is to the right. It's right here, so that's to the left. Put the CH2OH here. Wow, there it is. What would the Haworth look like for that? Well, the CH2OH would be down. And I'm going to just write OH, the CH2OH there, that way. The OH would be up. This OH is up. Left is above. Right is below. Right is below. And what goes here? Well, the two hydrogens. In a line formula, I leave that blank. This molecule would be beta-D-Fructofuranoside. See if I can write that in tiny. Beta-D-Ribopyranose. Beta-D-Fructofuranoside. Beta-D-Fructofuranoside makes up 57% of what's observed in aqueous solution. I could switch that around, that anomeric carbon around, and if I did, I'll just draw the Haworth. I would get the CH2OH up, the OH down, and then left is above, right is below, right is below. Here's alpha D, Fructofuranoside. Who is present at 3%. Okay, 3% for this guy. And so you see that we have Furanose rings in conjunction with Pyranose rings in equilibrium with fructose. So the fructose situation is a little more complicated. You should be able to draw the Fishers and Haworths of alpha D-fructofuranose, beta D-fructofuranose, beta D-fructopyranose, and alpha D-fructopyranose. You do not need to know the percentage compositions. You should be able to draw glycosides of both Pyranose rings and Furanose rings and name them. And so you need to be able to know and figure out what these structures are. And I went through in a previous Scribblecast showing you, if you're given a structure, how to break it up, convert it back to the Fisher. If you're given a structure like this, convert it back to its Fisher, and then open up the Fisher. Identify what the carbohydrate is, the D-fructose, and then go back and name it following our rules. Okay. Well, fructose certainly is an important sugar. One last sugar I do want to talk about in relation to what we've done with fructose and the fact that we get Furanose rings is a very important sugar, D-ribose, a pentose sugar. D-ribose is the pentose sugar that has all the OHs on the same side. D-ribose can also cyclize to form Furanose rings. Carbon 1, carbon 2, carbon 3, carbon 4. 4 atoms plus the oxygen makes 5. If it's cyclized, the fissure, I would get this. And if I was to produce beta, the OH would be opposite the reference. This is the anomeric carbon right here. I'd get CH2OH. If I was to draw the Haworth, the anomeric for beta, the OH is on the left in the fissure, so it's up. Right is below. Right is below. And the CH2OH is up. What you see here is beta-D-Ribofuranose. Beta-D-Ribofuranose is the backbone unit used in RNA, ribonucleic acids. Very biologically important. In DNA, we deoxygenate. We remove the OH at carbon 2. Remember, anomeric carbons, carbon 1, carbon 2. We deoxygenate this. And the Haworth would look like this. This molecule would be called 2-deoxy to indicate the oxygen-containing hydroxyl has been removed. 2-deoxy beta-D-Ribofuranose. Okay, so beta-D-Ribofuranose. Now notice, and this is just a point of interest, deoxygenation, so this guy was D-ribose. So there's D-ribose. Let's look at D-arabinose. D-arabinose has the second OH to the right. So this is D-arabinose. Notice if you deoxygenated D-ribose, remove that oxygen. Okay, so there's a carbon there with 2 hydrogens. And you deoxygenated D-arabinose, so 2-deoxy. Notice that 2-deoxy D-ribose and 2-deoxy D-arabinose become the same molecule. So what's interesting is 2-deoxy beta-D-Ribofuranose could also be called 2-deoxy beta-D-Ribopyranose. However, arabinose is not used in RNA, and hence, since 2-deoxy beta-D-Ribofuranose is used in DNA, it's thought to have come from RNA, and hence the ribose name is what is used. But just an interesting point that arabinose, the difference between arabinose and ribose disappears if you deoxygenate carbon 2. One last point is ribose sugars can also form Pyranose rings. And why don't we try drawing one of those? If I wanted to take ribose and produce a 6-atom ring, one of the atoms being oxygen, which hydroxyl group would have to cyclize? So let's put my OH back in. Well, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, to get a 6-atom ring with an oxygen, I'd have to use the oxygen of the CH2OH. I'd have to. Fun times. Cyclize. And the aldehyde would become, if I wanted beta, the OH would be to the left. If I was to draw a Haworth of this, beta, left is above, right is below, right is below, right is below, and there's nothing here because that's a CH2. Well, this I've drawn is beta-D-Ribofuranose. If we wanted to make the 2-deoxy, I would just remove the hydroxyl from carbon 2, and that would be 2-deoxy beta-D-Ribopyranose. Now, being the curious creatures we are, we like asking questions such as, why was 2-deoxy beta-D-Ribofuranose used in the construction of DNA? Why not use the Pyranose ring? Why not deoxygenate beta-D-Ribopyranose? Why, for example, did we not use glucose, which is the most abundant carbohydrate in the biosphere? Why did we not use 2-deoxy beta-D-Glucopyranose? Interesting questions. And there are people attempting to answer these questions by studying the chemistry of the ribopyranoses and the glucopyranoses rings in conjunction with the structures of DNA and RNA. Well, I think that's enough for now. I'm Dr. Brian Lloyd. This is my Scribblecast. Thank you very much.